Hardy-Weinberg Law of Equilibrium and microbreeding stand as two distinct and divergent methods of breeding, each with unique goals and varied applications. The Hardy-Weinberg Law of Equilibrium is a mathematical model that predicts the frequencies of different alleles in a population over generations. It assumes that the population is large, random mating is occurring, there is no mutation, migration, or selection, and the population is in equilibrium. This model can determine whether a population is evolving and estimate the frequencies of different alleles. On the other hand, microbreeding is a method of breeding that involves selecting and crossing individual plants based on specific traits or characteristics, intending to create new strains or cultivars. This method involves careful selection of individual plants that exhibit desirable traits such as high yield, potency, or disease resistance, and then crossing them to create offspring with these traits. While the Hardy-Weinberg Law of Equilibrium is a theoretical model used to predict the frequencies of different alleles in a population over time, microbreeding is a practical method to select and propagate desirable traits in individual plants. Both methods can be used in cannabis breeding, but have different applications and goals. The Hardy-Weinberg Law of Equilibrium can help determine a population's genetic diversity, identify the presence of genetic drift, and estimate allele frequencies. Conversely, microbreeding helps develop new strains or cultivars with desirable traits and characteristics for growers and consumers. Overall, the choice between Hardy-Weinberg breeding and microbreeding will depend on the specific goals of the breeding program and the resources available to the breeder. Both methods can be effective in cannabis breeding, but have different applications and require different expertise and resources. To expand on the differences between Hardy-Weinberg breeding and microbreeding, Hardy-Weinberg breeding is a theoretical model based on assumptions about a population's genetic makeup and behavior. For example, the model assumes that the population is large, random mating is occurring, no mutation, migration, or selection, and the population is in equilibrium. Under these assumptions, the model predicts the frequencies of different alleles in the population over time. Hardy-Weinberg breeding helps understand the genetic diversity of a population, estimate allele frequencies, and detect the presence of genetic drift or other evolutionary forces. However, there are more practical methods for selecting and propagating desirable traits. Microbreeding, on the other hand, is a practical method of breeding that involves selecting individual plants based on specific traits or characteristics. This method creates new strains or cultivars that exhibit desirable traits, such as high yield, potency, or disease resistance. Microbreeding involves crossing individual plants with desirable traits, selecting the offspring that exhibit the desired traits and then crossing those offspring to create a new strain or cultivar with the desired traits. One advantage of microbreeding is that it allows breeders to target desirable traits for growers and consumers. For example, a breeder might select plants resistant to pests or diseases, or produce high yields or levels of THC or CBD. Microbreeding also allows breeders to create new strains or cultivars that are unique and distinct from existing varieties. Overall, both Hardy-Weinberg and microbreeding have advantages and disadvantages, and both can be useful in cannabis breeding. However, the choice between the two methods will depend on the specific goals of the breeding program, the resources available to the breeder, and the level of expertise required to implement each method.